الله الرحمن الرحيم. I would like to welcome everybody to the lecture of the digestive system part 3. The small intestine has features. First, composed of the duodenum, jejunum, and the ileum. Also by the presence of villi, the glands of Rectum, which open between the intestinal villi, the gradual appearance of the goblet cells in the lining epithelium, the presence of plicae circularis, which are the Kerckering valves, which are the permanent folds present on the surface of the mucosa of the small intestine. On this diagram, we'll see this villi as small, very small, minute elevations or protrusions, magnified on the right side of this diagram. If we magnify this villus further, we'll see this villus containing connective tissue core in the center, and then it is covered with the tall simple columnar epithelium, and few goblet cells are present in between. We'll find also a layer of the muscularis mucosa, which is a smooth muscle layer within the mucosa of the small intestine. And then next on the submucosa is a connective tissue. This connective tissue, if we see it in the duodenum, the duodenum will find it as what is called the Brunner's gland in the submucosa of the duodenum. The muscularis externa, or sometimes we call it the musculosa, is composed of inner circular layer of smooth muscle and external longitudinal layer of smooth muscle. Has got a myenteric plexus or Auerbach's plexus between the inner circular and outer longitudinal layer, and the serosa layer, which is a connective tissue covered with a peritoneal covering. The villi, as we see in this nice diagram, we see the model of the villi on the right side, showing plexus of capillaries in the center of the villus, also blood vessel, lymphatic vessels, which help in the absorption of lipids in on the small intestine. Also, we see the simple columnar epithelial cells together with the goblet cells lining the villi of the small intestine. Here, the true vision, the true picture, the true figure we see under the microscope. We find the central core of connective tissue, simple columnar epithelial layer, many cells with few goblet cells. This is a typical villus of the duodenum. Now we'll explore the layers of the intestinal wall. First, the mucosa, composed of simple columnar epithelium plus goblet cells, connective tissue corium, as we mentioned before, the muscularis mucosa, the muscle layer of the mucosa, the glands of Rectum, which open between the intestinal villi, and then number 2, the submucosa, which is composed of connective tissue of variable thickness, the Brunner's gland, which are present in the duodenum only, and the Peyer's patches. These are lymphoid follicles present only in the ileum. We'll watch it later in the other sections. The muscle layer is called the musculosa or muscularis externa, as we said before, is continuous with the inner circular and outer, formed of continuous inner circular and outer longitudinal layers. The serosa is an outer the outer layer of connective tissue. It is called serosa if it has a peritoneal covering. If there is no peritoneal covering, it is called adventitia. The small intestine in this table is summarized to distinguish the main difference between each of its parts. The duodenal villi are broad and leaf-like in shape. The jejunum has got long and cylindrical villi like those of the ileum. The Brunner's gland is only present in the duodenum, but absent in the jejunum and in the ileum. No Brunner's gland are only visible in the submucosa of the duodenum. The goblet cells are only few in the duodenum, increasing in number in the jejunum and maximum in the ileum, while Peyer's patches, the lymphoid follicle that we mentioned before, is absent in the duodenum, absent in the jejunum, and only present in the ileum. In this slide, we'll see the major differences between the three parts of the small intestine: the duodenum, the jejunum, and the ileum. See, the sections of the duodenum have the major feature of the duodenum, which is the presence of the Brunner's gland. The Brunner's gland are here. You see this acini of the Brunner's gland. And then you watch the villi of the jejunum, which is long and cylindrical. While in the third part of the small intestine, we'll see the Peyer's patches in the ileum, which are large lymphoid follicles, characteristic of their presence is characteristic of the ileum. The mucosal cells of the small intestine is summarized on this table. We now will enumerate and describe each of the cells in the lining cells of the small intestine. We'll talk about the simple columnar absorptive cells. These are tall columnar cells with oval nuclei, basophilic base. The apex of these cells has got a brush border due to the presence of microvilli. Each cell has got about 3,000 microvilli. The main function of this cell is the absorption of food. The other cells are the goblet cells found in between the absorptive cells. They increase in number toward the ileum, start with few, moderate, and maximum number in the ileum. They secrete mucus for protection and for lubrication. Also, we'll find this characteristic cell of the small intestine only. It is the Paneth cells. They are present in the basal portion of the glands. The apex contain secretory granules which contain the lysozyme. The lysozyme kills bacteria and controls the intestinal flora. The intestinal flora are the normal bacteria which is present in the normal intestine. Also, we'll find the M cells, the microfold cells. The microfold cells are small, are the cells that cover the lymphatic follicles, the Peyer's patches, seen on the surface of Peyer's patches. It has membranous invaginations and no basement membrane under the M cells is seen. The function of these cells is an APC or antigen presenting cells. They present the antigen to the T helper cells. The GALT is the gut-associated lymphatic tissue. Abbreviation of GALT, the gut-associated lymphatic tissue, which is the which we mean those of Peyer's patches. The endocrine cells are described in the next table. On this table, we'll mention the enteroendocrine cells, which is present every now and then in the lining mucosa of the digestive tract. The G cells or G. pylorus cells secretes gastrin, and this helps in the stimulation of the gastric acid secretion. The S cells in the small intestine secretes secretin, which leads to stimulation of the pancreatic biliary bicarbonate and water secretion, inhibition of the gastric acid secretion. The K cells in the duodenum and the jejunum, they secrete gastric inhibitory polypeptide, which inhibit the gastric acid secretion. The L cells in the ileum and the colon secretes glucagon-like peptide, which stimulate the insulin release and also inhibit the hunger and the gastric acid secretion. The I cell in the small intestine secretes cholecystokinin, which leads to pancreatic acid secretion, gallbladder contraction, and inhibit the gastric acid secretion. The D cell in the gut secretes somatostatin that inhibit other cells of the diffuse enteroendocrine system. The MO cells of the small intestine secretes motilin that increase the gut motility. The EC cells of the stomach and the intestine secrete serotonin and substance P, which lead to increased gut motility. The N cells of the ileum secrete neurotensin, which inhibit the gastric acid secretion. Now we'll talk about the Pyloro-Duodenal Junction. In this junction, we'll find it the transition between the mucosa of the stomach, the pylorus of the stomach, and then the beginning of the duodenum. What happens here? We see a simple columnar epithelium of the stomach change into that of the duodenum, showing striated brush border with microvilli and goblet cells. Also, the gastric gland disappear. The gastric gland and the pits disappear, followed by the appearance of duodenal villi. The muscularis mucosa of the pylorus continues with that of the duodenum unchanged. See this nice view. The pits of the stomach here disappear, and we see the beginning of the duodenum here. The villi starts here on the duodenum. This side is the pylorus, and this side is the duodenum. And these are the Brunner's gland in the submucosa of the duodenum. This is the characteristic feature of the duodenum. Also, the Brunner's gland, as we mentioned here in the duodenum, start in the duodenum. The thick pyloric mucosa, the sphincter musculosa of the sphincter, the thick pyloric musculosa, the sphincter of the stomach, continues as a thinner layer in the duodenum. The serosa of the stomach continues unchanged in the duodenum. Now we'll start talking about the large intestine. The large intestine, as I'd like to distinguish each part, to mention the major features of each part. This is very useful for every student to know what distinguish the large intestine from other parts of the digestive tract. Number one, the villi disappear. No villi in the large intestine. Also, we'll find glands. Instead of villi, we'll find only glands. The crypts, sometimes we call it crypts of the large intestine. Number three, marked increase in the goblet cells. Number three, the tiny coli, which are the external longitudinal muscle layer, which are these are the longitudinal muscle layer forming bands called the tiny coli on the surface, the external surface of the large intestine. Also, the presence of appendices epiploiky, which are the fatty protrusions, yellowish fatty protrusions on the surface of the large intestine. Now, better view, larger view. You see this tiny coli. These are the external longitudinal layer of the muscle layer, forming three bands only on the surface, visible on the surface of large intestine. We see this appendix protruding from the cecum, the first part of the large intestine, the cecum, the ascending colon, the transverse colon. The large intestine generally has functions: the final water and gas absorption. It also helps in the secretion of mucus, lubrication, covering bacteria, cover the lubrication and the removal, cleaning of the surface, the production and storage of fecal mass. These are the major functions of large intestine. See this small, low power magnification of large intestine. The mucosa has no villi, as we just mentioned. The submucosa is just only connective tissue. The next layer is the muscle layer, composed of inner circular layer, and the tiny coli are only three bands of the external longitudinal fibers composing the tiny coli. Only in certain area we find this tiny coli, as we just watched it in the previous slide. The large intestine, as now we'll talk about each part, has a mucosa. The mucosa we just mentioned has another distinguished feature, the tiny coli, the major bands here and here and here, the tiny coli. The submucosa is composed of loose areolar connective tissue. The muscle layer, inner circular and outer longitudinal layer, forming three bands called the tiny coli. Here and here, see this? And finally, the serosa being characterized by the presence of small fatty protrusions called appendices epiploiky. Now we'll talk about the appendix. The appendix, we saw it as a small protrusion coming from the cecum. It has features like relatively small, narrow, irregular lumen. The abundant lymphoid follicle. Yes, we'll see many of these follicles on the we see it in the appendix. Every now and then we see many follicles here and diffuse lymphoid tissue, generally similar in structure to the large intestine, but contains fewer and shorter intestinal glands. It is also characterized by the absence, no tinea coli. The rectum and the anal canal. The rectum is similar to the colon, but has deeper glands or crypts. The anal canal shows a longitudinal folds, called columns of Morgagni. These columns meet each other. These are the columns of Morgagni, or the anal columns, meet each other, forming the anal valves. Here it is called the anal valves, behind which there are the anal sinuses. The anal sinuses here in this area. The rectum and the anal canal has a mucosa this mucosa is lined by simple columnar epithelium above what is called the pectinate line. Pectinate line is here. Above this line we find simple cuboidal epithelium. Below this line we find the stratified squamous non-keratinized epithelium below this pectinate line. Then finally we find stratified squamous keratinized epithelium at the anal orifice. Outside or near the orifice we'll find it stratified squamous keratinized epithelium. The lamina propria of the anal canal has anal and circumanal glands. Also we'll talk about the sphincters. Very important. The internal anal sphincter, which is formed by the inner circular layer of the muscle layer of the anal canal. These are smooth muscles that controlled involuntarily, while the pelvic floor has the levator ani muscle and the other skeletal muscle here around the anal canal. These are the external anal sphincter, skeletal muscle of the pelvic floor that works voluntarily. Okay, and also we'll talk now about the submucosa. The submucosa has two important features: the internal hemorrhoidal plexus and the external hemorrhoidal plexus. Both are of surgical importance for us to know. The internal hemorrhoidal is above, while the external hemorrhoidal is below the internal. The musculosa has two layers, inner and outer layers. The inner layer forms the internal anal sphincter, as we said before, the involuntary one, while the skeletal muscle of the pelvic floor forms the external anal sphincter, the voluntary one. Thank you for watching.